Well, hello, AP Bio students. It is Mrs. Shelton coming to you live from my kitchen once again. I'm just going to record a quick little screencastify on carbohydrates and lipids for you, just for those students who are not able to attend our Zoom meeting in order to get this discussion. Again, it's not necessary that you watch it. You might be able to just watch the videos and get it, but some people like hearing from me and kind of getting my explanation of the topic. So what we're actually covering and in Unit 1 is really kind of the rest of Unit 1. We have to cover carbohydrates, lipids, proteins, and nucleic acids. And so that is actually what the rest of Unit 1 is going to be about, is understanding basic biochemistry and understanding more specifics about those 4 different macromolecules. Now, when it comes to any macromolecule, what's really important is that you recognize that, first of all, a macromolecule is a very large organic molecule. When we talk about organic chemistry or organic molecules are always molecules that contain carbon. Remember that carbon can actually form 4 bonds, so that makes it a really unique structure. All living things then rely on these organic molecules. This is again, our carbohydrates, our lipids, our proteins, our nucleic acids. And it used to be thought that only living things could make organic molecules, but we actually can manufacture and make organic molecules in a lab. They behave and operate under basic chemistry rules and guidelines as well. So they just happen to be really large and complicated because of this carbon that's the backbone of all organic molecules that can form those 4 bonds. Now, also, when we talk about carbon-based organic molecules, carbon is always then going to be bonded with hydrogen. Hydrogen is going to satisfy bonding rules. So one of our basic rules for any organic molecules is that it has to have carbon and hydrogen in it to be classified as organic versus an inorganic molecule. So actually something like carbon dioxide, that's just CO2, is not classified as an organic molecule. It's an inorganic molecule is what it is. Now these elements here, so this is schnapps, or I also like to rearrange it to chop an S. These are the basic elements of living things, mainly carbon, hydrogen, and oxygen, but then we're going to have nitrogen and phosphorus in our proteins and our nucleic acids, and then proteins are also going to have sulfur, all of these. Now, our carbohydrates and lipids are only going to have carbon, hydrogen, and oxygen is what they're going to have in them. Now, when we do talk about macromolecules, what I'm asking you guys to do in your activities, these are summaries, you're kind of making summary charts like this. This is just really an overview to just remind you of our 4 basic macromolecules of lipids, carbohydrates, nucleic acids, and proteins. Next lesson will be nucleic acids and proteins, and then we'll also cover it again when we cover chapter 3. It's pretty important that we understand those. Now, with biological molecules, what we'll also address more of in chapter 3 is we'll talk about how these macromolecules start at a monomer, this single basic piece, and it's different. And carbohydrates have your monosaccharide as the basic monomer. A lipid then has glycerol and fatty acids as the basic monomer. Proteins have amino acids, the basic monomer. Monomer is the single repeating unit. And then the polymer is the long chain of all of those stuck together. Well, regardless of whether we're talking about a carb, lipid, protein, or nucleic acid, all monomers are going to join together using the chemical reaction called dehydration synthesis. We'll talk more about that when we get to chapter 3. And all polymers get broken down into basic monomers using the process of hydrolysis, where we have to return water back to the molecule to satisfy bonding rules. So we'll talk about those as far as how we join monomers and how we separate polymers more in chapter 3. What I want to get to, where there's a lot of detail, is lots of properties, functions, structure of all of these biological macromolecules. So where you should get to a point where you see a picture like this at the end of this unit number 1 and go, yep, that's a monosaccharide. That's probably a glucose molecule. Yep, that's a polysaccharide because it's long chains of these hexagon structures stuck together. You see this and you're going to go, yeah, that's a triglyceride. So we need to be able to recognize when we see the structure of a macromolecule, recognize how we categorize it, and be able to identify some basic properties about those macromolecules. Now, as far as functions are concerned, what we need to make sure that we are recognizing, so for today's lesson, we need to identify function-wise carbohydrates. So this would be your example of carbohydrates. We know carbohydrates as our sugars. So we know that that's our energy molecule that we utilize, but we also need to know that it's long-term energy storage like starch in a plant. We also need to know that carbohydrates form structure like in cellulose of a plant or also in chitin. Looks like chitin when you see it written out, but chitin in exoskeleton of insects as well. So there's a variety of functions in carbohydrates, but basically it's energy, energy storage, and then also structure. When it comes to lipids, lipids are going to be long-term energy storage as their primary function. However, the absolute most important function of a lipid that we're going to have to know in AP Bio is the formation of the lipid bilayer of all membranes. So all cell membranes have a lipid bilayer of phospholipids is the specific type of polymer of lipids that we're going to talk about. But then also lipids also include steroids, which are your hormone, which are a class of hormone molecule as well. Now, when it comes to carbohydrates, so this is just some basic information I need you guys to know about the structure of any type of carbohydrate. Now, carbohydrates are composed of carbon, hydrogen, and oxygen in a 1 to 2 to 1 ratio. These are the basic monosaccharides. Now, I say 1 to 2 to 1 because you guys probably know glucose is C6H12O6. Well, there's 3 carbon molecules, there's 5 carbon molecules of monosaccharides. We're going to learn about pentose sugars when we talk about DNA and RNA that have 5 carbons in them, but those are the monosaccharides. Now, they can be in a long chain structure. This is how you're going to see them. They're essentially where you don't see a letter. In organic chemistry, because there's so many carbons, they just eliminate the carbons. Wherever 2 lines join, assume that there's a carbon there, is what you're assuming in any organic chemistry model that you're seeing. So this would be 6 carbons going down, but we're seeing a hydrogen and a hydroxyl group attached to each of those central carbons. Now, in a water environment, what actually happens is this is going to form into a ring structure. So this is why we often see glucose just written in these little hexagons. This oxygen up here ends up being the one of the oxygens end up being one of the bonds that ends up connecting these. So the ring structure is what I want you guys to be able to recognize. So in something like this, here's the ring structure of glucose. Here then is a disaccharide. If I join 2 monosaccharides together through dehydration synthesis, I now have a disaccharide. Sucrose, common table sugar, is a disaccharide. It's actually glucose and then fructose. And you can even see fructose has a different shape to it, is what's there. Now, I'm asking you in today's assignment to put a picture of lactose. Lactose is another disaccharide for you guys to see. Now, here then is a polysaccharide. And polysaccharides are just long chains of monosaccharides that have been joined together through dehydration synthesis reactions. And they vary in length, they vary in which monosaccharide is linked. They also vary in where the bonding is as well. And that's what makes differences in, say, things like cellulose versus then a starch versus glycogen. And so those are some polysaccharides you're going to have to look up. But they're all long chains of glucose, but how they're bonded together, are they in branching, or are they in long chains? Is it an alpha to beta or 1 to 4 linkage? We'll talk about that in chapter 3. They all make differences then in the ultimate structure and function then of these polysaccharides. So here's just a quick example too of starch, which is a glucose polysaccharide. This is actually a plant's long-term energy storage molecule. Glycogen is, again, long chains of carbohydrates. This is what we do when we end up having too much sugar in our blood sugar. That's actually what insulin tells your liver to do, is start binding up all those simple sugars into glycogen in order to maintain a proper blood sugar level. And you can see it's this branch pattern, so it's also easy for us to break down and utilize when our blood sugar drops low. Same thing with the starch in a potato. This branch pattern actually makes it easier for enzymes to get in, break down the simple sugars for aerobic respiration to take place. Now, cellulose, on the other hand, cellulose is this long, straight chain that is for plant structure. So plant cell walls are made out of cellulose. Your house is made out of cellulose. So all the wood that we have, that's all cellulose, and there's only a few bacteria on this planet that actually have enzymes that can break cellulose down. So this is why cellulose is your paper, cellulose is your house, cellulose is the box that my computer's sitting on right now. So, but it's this structure here of this long chain and also the way the bonds are formed here that animals just don't have an enzyme that can actually break this down and perform the hydrolysis process in order to break it down to simple sugars. Now, lipids, so we're going to talk about next. So lipids here are lipids. They're what we utilize for energy storage. Like I also told you, they're really important for cell membrane. We don't have cell membranes without lipids. And then also the other function of lipids is to serve as a steroid. So steroids being this lipid soluble hormone that is long distance signal throughout your body. Now, the basics of any lipid is there's going to be a glycerol molecule. So these are the monomers. A glycerol molecule is just a 3-carbon molecule with hydrogen and hydroxyl on it. And then fatty acids. So fatty acids are carbon, hydrogen, and oxygen also, but it's just this long hydrocarbon. And hydrocarbons are just carbon and hydrogen. That's all they are. And we'll learn the name of this functional group when we get to chapter 3. It's called a carboxyl group. But that's all a fatty acid is, are these long hydrocarbon chains with this carboxyl. And it's at this region where glycerol binds to fatty acids in order to form then what we call a triglyceride molecule. So if we're having olive oil on our salad, well, it's a form of triglyceride. If we are having the fat chunk on your steak, doesn't that sound appealing? Yummy. That's a triglyceride. So it's 3 fatty acids bound to 1 glycerol molecule in order to form that triglyceride. Now, these fatty acids, though, differ in how long the carbon chains are and also whether or not there are double bonds in that hydrocarbon chain. So this is something you absolutely need to recognize is the difference between a saturated fatty acid versus an unsaturated fatty acid. If it's a saturated fatty acid, all the carbons have single bonds, and this term saturated means that it is absolutely full of the maximum number of hydrogens that can attach to these carbons. Now, if it's unsaturated, that means there's at least 1 or more double bonds in the carbon chain. And when we have a double bond in the carbon chain, remember that carbon can only make 4 bonds. So in making 4 bonds, well, 2 of the bonds are satisfied here with the carbon-carbon double bond. So now we have less hydrogens in here. Now, the other thing that happens in an unsaturated fatty acid, remember molecules are 3-dimensional, this double bond actually causes this fatty acid to kink and bend. Well, that bend is actually really important in creating space between fatty acids and also creating space between other triglycerides. And so it actually makes it much harder for these lipids to solidify in cooler temperatures. So we'll actually see that this is something that plants actually do in order to maintain their cell membrane fluidity is they have more unsaturated fatty acids as part of their phospholipids for their cell membrane. Now, saturated fatty acids are typically what most animal fats are, is we just form these carbon-carbon single bonds, and this amount of this type of lipid, usually we find solid at room temperature. So it, which if you're a plant, you don't want solid plasma membranes at room temperature because then you can't get materials in and out of your cell. Now, as far as some of the different looks to some of our lipids, here's again another model of a triglyceride. Again, since it's just this long hydrocarbon chain in organic chemistry, oftentimes you don't see the hydrogens or the carbons written. They just show you where each bond between the carbon and hydrogen is. Now, this would be a phospholipid. Phospholipid is what our cell membranes are made up of. It's still a glycerol, but now it's 2 fatty acid chains, but now there's this charged phosphate group that's up here. And so this is where we get phospholipid. Well, we have to remember like dissolves like. Triglycerides, these are nonpolar molecules. And as a nonpolar molecule, this will not dissolve in water, which for a cell membrane structure, we have water and we have regions that are not water. So by adding this phospholipid onto the glycerol, we now have a region of a phospholipid that's actually referred to as amphipathic. it is both polar, because this is a charged portion that will dissolve in water, and then it also has this non-polar region that won't dissolve in water. And that's super important when it comes to formation of the lipid bilayer. Now, steroids have a very unique structure to them. It's actually 4 fused carbon rings, and then there's different functional groups that add to them to give different steroids. But these hydrocarbon chains, instead of being these little tails that stick out, they form these ring structures, is what ends up happening. So anytime you see this 4 ring structure that doesn't have any oxygens attached to it, it's just hydrocarbons linked around, that's going to be your steroid molecule, is what you're looking at. And then last picture here that I do have for you, this is just more detail then of a phospholipid. So you actually see the glycerol molecule. Here's a saturated fatty acid. Here's an unsaturated fatty acid. And then here's my phosphate group. This is typically the model, the simplified version you're going to see throughout our textbook of a phospholipid, where it's just showing this polar head region, this polar region that's the phosphate group that's going to dissolve in water, and then I'm going to have these 2 nonpolar tails. And they end up forming this lipid bilayer, and this is what our cell membrane is going to be focused on and or built out of, and we're going to see lots of this in upcoming chapters. For today, on today's assignment, however, what you need to make sure that you are understanding is the summary information I've asked you here. So carbohydrates, I've linked for you a YouTube video by Dr. Mike. It's very simple. His diagram that he has on the whiteboard is exactly what you guys need to know. The reading by Khan Academy on carbohydrates is super easy too. Khan Academy has tons of resources for AP biology students specifically. And then I ask you to summarize the information on monomers, on polymers, on some common monomers and polymers of carbohydrates and lipids on this document. So that is today's assignment. Hopefully this screencastify helped you out. And if you do need additional help, please email me or join in on a Zoom open office hours in order to ask help too. So with that, have a fantastic day, and I hope to see you guys again in class. Let's turn this off. Ta-da!